Now, this is the second of these science seminars. There's going to be one a month from now on into the, I don't know how long until they stop, but anyway, certainly for another 2 or 3 months. And the people who, well, what I'm going to do is, in the first 3 slides, show what you need to understand from the first webinar in order to understand this one. Because we dealt with a topic in the last webinar, which will be available on the publicly at some time soon, but it covered different things. It was mainly about the genetics of Cavernoma, but some of what was there we need today. And then the other thing to say is that we're about to have a new website, and there will be a blog on the website, which is the same topics that I'm talking about. And the blogs will always be after this webinar, seminar, whatever you call it, meeting, because last time I learned a lot about what people needed to know about from the webinar that went into the blog. So I hope you're okay so far. So the first slide proper is coming up, and this is a recap, and it's just to remind you that Cavernoma form, Cavernoma form in the blood capillaries, and the blood capillaries take the blood from the arteries through to the vein and back to the heart. And the blood capillaries are very tiny tubes. This is what one looks like in everything I do is diagrammatic, by the way. It looks a little bit like this. It's made up of cells in the cell wall, roughly this kind of shape in relationship to the diameter of the capillary. And that makes it a very weak structure because most other blood cells, like the arteries or the veins, have much more tissue surrounding them to give them strength. But these are very delicate structures because they pass oxygen from to your brain, very important, and nutrients to the brain as well. And so there's nothing much impeding those in this structure. There's a sheath around it to give it mechanical strength, but that doesn't do anything to stop any of the things, including red blood cells, passing out. And in cross section, you cut it, it looks like this. We're going to be talking about that sort of cross section quite a lot in today's talk. Now, 99.9% and more of people have what we will call normal blood capillaries. And the feature of those that matters is that they have a tight junction between the adjacent cells of the blood capillary. So in the borders between the blood cells, all the way along here, there are tight junctions, and those tight junctions prevent blood leaking out and such like. But if you have a cavernoma, and if you're in the 1.1% or rather less of the population that has a cavernoma, you've lost those tight junctions, and the cell can become leaky and leak out red blood cells, and it can enlarge. And we'll be talking about the structure of a cavernoma more realistically in a different webinar. That isn't really the topic of this one. And you're welcome to ask questions. Don't forget, I can't see you putting your hand up, but you have to unmute yourself and yell and say, David, stop. Now, the next part of last week's talk, or last month's talk rather, they're one a month, these talks, is to do with the genes and a little bit about genetics. Because I'm sure you are aware that there are 3 known what are called Cavernoma genes, named CCM1, CCM2 and CCM3. They have other names as well historically, but that's what we're going to call them. And so what we have, and genes sit on the chromosome, so here's a mock chromosome with a gene on, and this is one of these Cavernoma genes. But an essential feature of understanding the genetics of Cavernoma is to know that although you have this gene, it can have different forms. And if you are one of the 99.9%, the form that you have, I'm calling the tight junction form. So the tight junction form is what most people have, but if you have any form of Cavernoma, the genes, well, if you have a familial Cavernoma that's inherited, you will inherit one or other of these genes, CCM1, CCM2, or CCM3. And the form of the Cavernoma gene that you have will be what I'm calling the Cavernoma or CCM form, the mutant form. This is, as far as as far as having a cavernoma is concerned, if you have a familial cavernoma, that's straightforward. But if you don't, if you have what is known as a sporadic form, it's not inherited. Nonetheless, by the time you get a cavernoma, the form of this gene is the mutant form. And that's what we talked about last time. I shan't talk about it this time unless if you ask questions at the end. So, just to recap this slide, 99.9% of the population have the tight junction form of the cavernoma gene, and in the less than 0.1%, you have the cavernoma form of one or more, or one really, I don't think it would ever be more, of the CCM1, 2 or 3 gene. Okay, so far. Great. I take you, I can see you, Susan. I use you as the talisman. So, next recap is that what genes do is they are the code for the cell to make proteins. They're the information provided for the cell to make a protein. And I've coloured genes in blue here and proteins in yellow. So if you're one of the 99.9% of people, you have the tight junction form of your genes, and you produce a protein, CCM1, CCM2, or CCM3, for each of those genes. And these are lowercase, whereas the genes are uppercase. It gets a little bit confusing, I realise, but the yellow is protein, little CCM1, CCM2, and CCM3. And a vital part of this whole story is that those proteins combine together to form a complex. And the complex has CCM2 in the middle, and it joins, as it were, CCM3 and CCM1. This is known as the adapter complex in the literature. I've called it that. I'll probably call it the control protein, the control complex, sometimes in the talk. If you have the cavernoma form of the gene, and this applies to the 0.1% of you with a cavernoma, then you don't have a complex. And you don't have a complex whether it's the CCM1 or the CCM2 or the CCM3 gene that you have. Any one of those, being the cavernoma form, is sufficient to stop the complex forming. And that's true whether you have the familial form or the sporadic form by the time you get to the cavernoma. I'll ask questions about the difference between familial and sporadic if you wish to, or you can look at the blog when it goes live shortly. And so, how many of you recognise that? I think you need to show Pat and she will tell me. No one recognises that. That gives one bit of proof, scientific proof in this meeting, and that is that I'm older than the rest of you, because that is the fat controller for Thomas the Tank Engine. I don't know how many of you even know about Thomas the Tank Engine, but I used to read Thomas the Tank Engine stories to my children, and the fat controller controlled the fat engines. He was the one who was the Gauleiter of it all. And so I'm drawn him there because that is what the complex does for us. Now, this is where the talk today starts and being different from that from last week. And what this slide shows, and down at the bottom right here, is a cross section through the blood capillary. So you have to imagine that it goes all the way around in a tube, and the inside contains blood. Was that a question? No? And so what you have at the bottom here is cell 1 and cell 2 joining with joining like this, and that's what you see in the top part of the diagram here. It's just a little bit of join between cell 1 and cell 2. So you have blood in the middle flowing along here with its blood corpuscles, and you have the brain tissue getting the oxygen and the nutrients from the brain down here. What is on this slide is everything you need to know about today's talk. It's not the totality of what you get in the cells by any means. But the first component is easy. That's the controller complex, the adapter complex, and that's there because if you're one of the 99.9% of the population, you have the tight junction genes, which give rise to this complex. What happens is that the CCM1 protein here links to, before I do that, I should have talked about this molecule in red. This is another protein. This is an important protein. This is called aderin, and each cell has aderin molecules with a tail inside the cell, a bit that crosses the cell membrane. This black line here is the cell membrane of capillary cell 1, and then it has in the space between the cells 6 units which combine and which form what I'm trying to get. They join. Thank you. They join with these 6 elements from the cell from the other side, and so what you have then is an aderin molecule, which is the red bit and the orange bit, linked to the aderin from the adjacent cell, and this holds these 2 cells together. And in order for that to happen, you need a link, and that's what I showed too quickly, between the CCM1 protein, which is a tight junction protein, if you like, that links to the aderin molecule. It doesn't link directly. C1 doesn't link to the aderin directly. It links through a series of other proteins here that don't matter for today's discussion. It's simply that the link here is the crucial part of this talk. And that we're calling a normal aderin junction. Now, actually, we really mean it's the normal tight junction, because in addition to this link here with the aderin molecule, aderin, because they adhere to each other, very subtle biochemists, you have tight junction proteins up here which form the tight junction. And so I'm going to think of, or you're going to think of, I hope, this whole here as being the tight junction, the aderin and the tight junction molecules together. Are we okay so far? I take silence to mean yes. You have to put your unmute on and yell no. We're not doing very well for questions so far, I might say. Okay, so that's what you are. That's what you have if you're the 99.9%. If you're the 0.1%, then one or more of your genes, by the time it gets up to here, is a CCM gene. You have to have 2 CCM genes in each cell, or you do have. If you're born with a familial form, one of them is already CCM, and you get another one during your lifetime. I'll explain that later if you want. But the upshot is you have no complex. That's true whether you're a familial person or a sporadic person.
I mean, the upshot is you have a cavernoma, isn't it?
And the upshot is you have a cavernoma. The upshot,
David,
well, the upshot can be you have a cavernoma. You have to have this to have a cavernoma. You don't necessarily have a cavernoma, I think, and we'll talk about that in the next talk, if you don't have the complex. That's a subtlety too far for this talk.
David.
Hello?
Hello, yes, question. So to be, for want of the phrase you're using, to be normal, you have all 3 of these CCM numbered things, 1, 2, and 3.
Yes.
To be us, or the carer of someone like us, so to be the sufferer, you have 1 missing of those 3, so you don't make a complex, or 1 damaged of those 3, or 1. You have more than 1 missing. If you're a carrier in the way that you're talking about, Judith, one of those CCMs is a mutant. Now, it can be mutant in different ways. If we're going to give a strict answer to the question, it can be any of the things you say, and in fact, there isn't one single mutant of any one of those genes. There are various ways you could have mutants, but they all have the same effect. They then don't allow that protein to form, and you don't get the complex. Right, so you have to have all 3, they have to link together, and they all have to be normal versions of those 3.
That's correct, and that's what 99.9 something % of the population have.
Okay, great. Thank you very much for asking that, Judith. It's very nice to have questions from anybody when you don't understand something or want to ask a further.
Yeah, it was just for clarity. So yep, great, thank you.
Absolutely, and that's any of you should do that. So if you don't have the complex, as you don't have here, you no longer get any join to the adherin, and the adherin has a different structure. Then you no longer get these units binding to each other, and so they separate, and the cells can then separate, because at the same time, the tight junction proteins that link here are torn apart, and so you have a gap down here. So not having the complex means the adherin takes this abnormal form, the 0.1% of the population, rather than the normal form that you had when you have, as Judith was asking, all three present there. What happens is that one of the proteins down this link here links in to bind to the adherin, and it has to bind in a particular way to allow that form to take place. So we're calling that the non-functioning adherin junction, or the non-functioning tight junction. It's what we've called the Cavernoma junction. And when you have that, then blood can leak through from the bloodstream through to the brain, because there's now no longer the tight junction, there's a gap here, and so blood can get through. Now, blood can get through in two ways. In the normal course of event, blood can ooze from the blood in the capillary through to the brain, and it does so, I don't think we know whether it does so continuously, but it can certainly happen a lot when you're not aware of it. That is in distinction to when you have what we call a hemorrhage, which is something much more dramatic and which happens at unpredictable intervals to people, maybe a year or two apart. And when that happens, the cavernoma gets larger normally, and the symptoms that you have can get worse. So we need to distinguish between what I'm calling oozing blood, which is what we've got here, and a blood hemorrhage, which is a more dramatic occasion. You can ooze blood more or less continuously, but the hemorrhage is a dramatic, well, it's dramatic, it's a thing which happens occasionally. Now, we don't know what triggers the hemorrhage. So what I'm talking about here is the leakage of blood. And when I'll talk more about hemorrhage, not in this talk, but in another talk, so that we can cover that. That's a very important feature, obviously, but it's not part of this talk because it's not yet properly understood.
David, can I interrupt and ask a question on behalf of someone?
Sure.
I saw a talk where Dr. Awad said that at the 3 genes have to work initially to form a heart in the embryo, and then they deviate. Does that chime with you? That Awad said that 3 genes have to work initially to form.
That's right, and that's a different. I mean, these genes don't only work in Cavernoma. And so the genes can be active in different ways in different cells for different functions. And I don't know that I don't know sufficient about that to answer that. But the genes are present in all the cells of your body, not just in your blood capillary cells, and so they have other effects. Is that okay? I'll take it's okay unless you come back to me. Okay, so I'm drawing this picture here. This is for the people with the Cavernoma. What we've got here is an explanation of the tight junction loss in that first slide. And so this is just an extra description, if you like, of what was described very simply in slide 1. Then that's, in a sense, the end of most of this talk, except we haven't talked about this receptor up here, which I drew. And that's really an introduction to the talk next month. And the talk next month is about the microbiome and the way that the microbiome has an effect on the Cavernoma. And it's all to do, this is a preview for next month, it's all to do with this receptor here. Now this receptor
well, receptors in cells have a bit of them outside the cell and a bit of them inside the cell. The bit on the outside will bind to something in the blood. And this particular receptor, this is a particular receptor, cells have lots of different types of receptor. This particular kind of receptor is known, well, we'll come across it next week, but it is specific to the substance, the lipopolysaccharide, that emanates from the microbiome in the gut, and it binds to this receptor here. And when this receptor here has that lipopolysaccharide bound to it, it changes the structure inside, and then that does something we'll talk about next month inside the cell. And that's a very important aspect to this story, but I thought it wasn't good to talk about it all in this week. So the receptor spans the cell membrane, a part outside, a part inside, and this is the way that something in the blood can influence what goes on inside the cell without actually getting into the cell itself. This lipopolysaccharide, or any of the substances in blood which link into the capillary cells, don't have to get into the lipopolysaccharide doesn't have to get into here to have its effect. It can do it indirectly. That reaches the end. So I'm going to stop showing slides for the moment, although we can come back to them if you wish to. At this point, you can ask questions, because although you're still being recorded, the way it gets made public won't include these questions. We decided we would make all the recordings record the whole. It enables me to go back and remember what people asked so that I can make the blog better than what it would have been without this talk. So it's over to you. Hands up if you vaguely understood it.
Yeah, vaguely.
Okay, well that's good. And those of you who didn't put your hand up, you can ask the questions.
Hi, David.
Hi.
Hi. I'm calling on behalf of my wife, who's currently in bed. She's in severe pain quite often. She's been diagnosed with 2 cystic cavernomas on the left side cerebella, on the rear of her head. Would a bleed cause any severe headaches, or would that be from something else?
I can't answer that question with high knowledge. The answer is almost certainly, though, yes. And she isn't alone in having that effect, even from within the cerebellum.
Right, great. Thank you very much.
David, I'm going to ask again. We know there's a difference between oozing and a hemorrhage, which I think we all understand that generally some people tend to have oozing, which is more like what I have, than a 1 or 2 off major bleeds. Are there any symptoms that are more likely if something's oozing? So I have uncontrolled epilepsy. Would we associate that more with an oozing situation than necessarily a 1-off or 2-off big bleeds? Do we know if the symptoms vary more so by the type of issues you have?
I'll answer it in the following way, and you can tell me I haven't really answered the question in a moment, Judith. The normal state of a cavernoma is to have the oozing form, and it doesn't necessarily ooze the whole time, which implies that those tight junctions, as it were, can be so tight as one another. But that's the normal form that happens, and the symptoms you get, whether they are epilepsy or whether they are other symptoms, because the cavernoma is in another part of the brain, those are, I don't think, going to be changed all that much. But again, I'm not a clinician by this state. What happens when you get the hemorrhage isn't only, the major thing you get when you get a hemorrhage is the enlargement of the cavernoma. And that's a different effect. And we shall talk about that when we'll talk about the way that cavernoma develop, because there's quite a lot now known about the way that cavernoma develop. What I've talked about is the changes that take place in a single cell. But in order for a cavernoma to develop, you then get the similar changes in adjacent cells. And it's a very interesting explanation as to what happens to get the cavernoma formed, because lots of cells change all in one vicinity compared to what I've talked about, which is just one cell change. But when they all change, then when you get a bleed, then you get a bigger effect of the whole. Does that help explain or not?
Yeah, it does. I recently read one of the letters from my consultant, and it says that they don't see any obvious signs of bleeding.
Right.
But I obviously get symptoms. You know, those who've heard me on previous talks will understand I get symptoms from my cavernoma, but the consultants think they haven't bled. So is that just because they exist and there's slight oozing? Because obviously if there's no major bleed, I don't have this kind of one-off.
Okay.
And the oozing doesn't seem to, I can see somebody else asking something similar. The oozing that I must have doesn't seem to show any difference on ongoing MRIs. They don't look any bigger, which is why the doctors say they're just not bleeding, or they don't believe there's any change, yet I still get a lot of symptoms. I don't know if I'm making enough sense.
You are, and I don't know that I know it well enough. I think when your clinician says about a bleed, he means the hemorrhage, the big hemorrhage. And I don't think he's talking about the minor effects. I don't think cavernoma ooze the whole time. They won't change shape. The major effects you get from the cavernoma aren't due to the oozing. They're due to the presence of something that's significantly sized in your brain to interact with the nervous tissue around.
So just the fact that they're there can cause the symptoms even without any big one-off event.
Yes. The big one-off event will change the size of the cavernoma and make the symptoms be different because of the interaction between some. If you've got a thing, the golf ball inside of your head, it has a bigger effect than if you have something that's only 1 millimeter in diameter.
David, I have another question for you.
Right?
Hold on, I have to pick it up here. Sorry. Right. I think this means the oozing, why the cavernomas show up on the MRI, even if they have not hemorrhaged.
The reason why cavernomas show up on MRI is not due to the oozing or the blood particularly, although you can, different forms of, we'll talk about MRI in another session. There are different forms of MRI which show up different things. What you see on an MRI is basically the structure of the cavernoma, and the extent to which you see any blood or any blood around there is going to be dependent on the type of MRI you have. But the main thing you see in an MRI is the overall structure of the cavernoma. It's the cells, not the blood particularly. I mean, you can see the blood in some forms of MRI.
And I hope I got the question right, who asked the question. So do re-ask if I got that wrong. And can I just pipe in? This really isn't a question, but I mean for others, I had a hard time. This is just to pipe in really with that nomenclature. I mean, David, you said adherin. What a great name biochemists gave that protein for a protein that adhered to itself. But I found very confusing that the CCM genes were for those who inherited a gene that would lead to a cavernoma that we call CCM. But then the protein, when it was not associated with the CCM gene, was called in lowercase a CCM protein. That was terribly confusing for me. But, once I got it. But it took me, I had to pause to catch up when you first gave me that talk. Anyway, that's that I did find confusing, but so be it.
I think that is confusing. I didn't
emphasize that during the talk, but you talk about Cavernoma genes, but of course most of the time they're not responsible for Cavernoma. It's only when they become mutant that they are.
Yeah.
That's why I called them tight junction genes. You don't find tight junction genes in the literature. They're peculiar to these talks.
Yeah. So, yeah.
I have one question regarding Heidi, regarding your last slide.
Shall I put it back up?
Yeah.
This one?
Yeah, the receptor. You mentioned that the microbiome, the bacteria in the gut, can influence the behavior of the cell. This means that the gut health is involved in the Cavernoma.
Absolutely, yes. This was a very exciting finding about 3 years ago only.
Okay.
And let me tell you the story about it, if I may, so it doesn't take that long to say. But there's somebody called Mark Kahn who works in Philadelphia on Cavernoma, and he's not particularly interested in the microbiome at all. In fact, he didn't even probably know about it. Well, that's not fair, but he wasn't interested in it. And he worked in a laboratory in one building, and he was made to move from that building to another building. And he worked on mice, because you have mice which can get Cavernoma. And he had in his animal house mice that got Cavernoma. And he had to move them all to the new building, and they no longer got Cavernoma. And so here was all his research that depended on mice getting Cavernoma, because then he could work on Cavernoma, suddenly didn't have them. And it turned out that the reason why they didn't get the Cavernoma in the new animal house was because the environment was different. Their gut, your gut has billions of bacteria in it of many different forms. And it was because the bacteria had a different form, had a different composition. There are many thousands of different types of bacteria in your gut, and each of us has a different set of bacteria. But it turned out that these mice didn't have the bacteria that were needed to form Cavernoma. And that opened up a whole new subject area. And that's really the subject of the talk I shall give next month. But so then the question was, which was cause and effect? Because at that point you didn't know. But a lot of work's been done on that since. It's a very exciting story. And in order to get a Cavernoma, you have to trigger this receptor up there with something from your gut.
Okay.
And so when I said that having having the lack of a complex, was that enough to cause a Cavernoma to form? It wasn't, because you have to have something from the gut as well.
Okay.
And that, of course, is a very exciting thing, because if you have to have something from the gut as well as having a Cavernoma gene, then maybe you can get some kind of drug which worries about the thing in the blood, not have to worry about the thing in the Cavernoma cell. So, 4 weeks' time, need to be back here.
Okay. Yeah, certainly. That's very interesting. Thank you.
I mean, it's one of the most exciting recent findings about Cavernoma.
But I guess this could imply that the type of food also could help.
Indeed, it does. And this is a big subject. And one thing that you get in food quite a lot is emulsifiers. And emulsifiers tend to let the wrong bacteria get through into the bloodstream. And so there's a whole food diet book available from Angioma Alliance, which is the American equivalent of Cavernoma Alliance, which you can use to how to eat healthily so as to reduce your impact of Cavernoma.
Okay, that's interesting.
It is.
David, excuse me, I have a couple questions for you.
Right.
Are you ready?
Yep.
Okay. You described a big hemorrhage as being more of a dramatic event than oozing blood. If that's the case, what's the difference between an AVM and a hemorrhage?
Oh, well, an AVM is a different type of structure altogether. And we can talk about that. It's often associated with cavernoma, but it's a different animal, if you like. So it's a different structure, and it works in a different way to cavernoma. Does that answer the question?
I'll see. Questioner, if you want more on AVMs, can you let us know? And we'll just go to the next question for the moment while we find out. Another question: how much blood is oozed out? For example, that is, what determines whether it's oozed and when it is a bleed? You know, a bona fide, well, not bona fide, a bigger bleed. And does the oozed blood become absorbed eventually?
I don't know the answer to the first part. I don't really know what makes oozing vary in size. I mean, if you believe what I've said today, it could all be the constant. So there's something more going on in a subtle way. What I've described is just basically the basics of a cavernoma and why blood can get out at all. The big bleed is a dramatic effect that I don't think is known about in the literature. I've not found that, and I've asked people who should know. So that's a different topic, and we'll have to answer that at a different time. But I think you'll find that you don't get a sort of constant oozing. What was the last part of the question? Oh, does it get absorbed? Yes, I mean, what happens to the blood is not a constant. It can get out from the cavernoma into the brain under some circumstances and stay within it in others. And I don't know more about it than to say more about it than that.
Okay, if I can pipe in a couple things, and then after I do, yes, please, more on AVM. But before I do, can I just say from the chat line, and I think she'd be happy for her name because she's saying, thank you, David. This is very informative. I have to go till next time. And that was to everyone. That was from Susan. So an adios from Susan. And Jana has put a link in if anybody's looking on the chat line or wants to put their chat line on. If you want to know more about the endothelial cell junction, there's a good scientific review article, and Jana has put a link there if you want to read more about it. And one more, another question besides a little more on, or maybe a lot more on AVM. How long does it take for the blood to be reabsorbed into the body, as there must be a difference between when it oozes and when it bleeds?
And I don't know the answer to that at all. I've not even seen an answer to that.
Okay, so without further ado, well, there may be some questions from the floor, as it were. Oh gosh, is it a floor? I don't know. Can you give more on AVM?
I think I was going to do a talk on AVMs at some stage. I think I'd rather not preempt that now. I need to do a bit more reading about them. What I could do is I'll send an email round with with something to fill up for a questionnaire. There's a little bit more about AVM on the Angioma Alliance website. If you go into Google or whatever you go into and type Angioma Alliance, you'll get to their website, and they have a page called Fast Facts or Questions. I forget exactly. It's very easy to find, and they have more about AVMs there. I'm not confident enough. I know sufficient to do. I'd rather do the preparation and talk about it. It'll be 3 or 4 months before that comes up. Well, maybe we've reached the end of that. And Deborah, you're in mute. Fine.
Yeah, I've unmuted. So I've got a quick question. So Mark's had a cavernoma that's been partially resected, and he's had symptoms again since that operation. Does that mean that the cavernoma has got bigger, or is it just the bit that's left has continued to ooze?
I don't think there's any way.
He was told that it hadn't actually bled, but it hadn't actually hemorrhaged, but he was having symptoms. But it was a bit like the other, I can't remember her name, who asked the question. What's the difference
My
between
question.
Yeah, Judith, that's it.
No problem, Deborah.
I don't think I could. I don't think I can answer that simply because you can't answer that simply from what you've said. You'd need to go back to the clinician and ask more about it.
Yeah.
I mean, I think if you haven't had all your cavernoma taken out, the bit that remains can still act like a cavernoma. The hope is that it will act much less like one because it'll be much smaller. But then the fate of that, you would need to learn from your clinician.
Okay. Yeah. Thank you.
Okay. I mean, I think that's probably everybody satisfied. Thank you very much for coming. I hope that was instructive. It was rather little content in some ways, only 2 major slides after the last week's talk. But very, very nice to see everybody. And we'll call it a day there and see you again, I hope. Thank you.
Thank you, David.
Thank you.
Thank you.
Bye, everybody.
Bye.
Bye.
Nice to see
Bye, everybody.
you.
Thank you.
Bye.
Nice to see you.